So welcome back. Whether you took a long break or just a short break and jumped across to the next section of the recording or of the presentation, welcome back. Here are our concept check questions. Remember, these are the questions that I left you with at the end of the last recording. Explain how oxygen isotopes and sulfur bacteria helped figure out the chemistry of photosynthesis. Well, remember, the sulfur bacteria carry out the chemosynthesis process, where H2S is serving the same function as H2O in photosynthesis, a source of electrons. And the H2S is being reduced to pure sulfur. And some of the electrons are then being used to reduce carbon dioxide to glucose. The remaining excess electrons are used to reduce oxygen to water. So water becomes a product of the chemosynthesis process. But more importantly, what it indicates is that the sulfur that is the direct product of the oxidation of H2S only comes from H2S. There's no other place that it could come from. And if the H2S is serving the same purpose as water in photosynthesis, then the water must be the direct origin of all the oxygen that's being produced, and only the water. Because if we look at the two reactants, there's carbon dioxide, which contains oxygen, and there's water, which contains oxygen. Either could be the source of the oxygen byproduct of photosynthesis. But the parallel to the purple sulfur bacteria suggested that the oxygen product of photosynthesis must come strictly from water. The problem from Van Nijl, the biologist who figured this out, was that he could not demonstrate it. But the oxygen isotope study demonstrates the reality of water being the sole source of oxygen. Because if you give carbon dioxide containing the oxygen 18 isotope to plants, none of the oxygen 18 turns up in the oxygen gas. But if you give oxygen 18 containing water to plants, then the oxygen gas contains the oxygen 18. And only if you give the oxygen 18 in the form of water molecules do you get the oxygen 18 coming out as the oxygen gas, confirming what Van Nijl had already suspected on the basis of what he'd seen with the sulfur bacteria. So it was a combination of Van Nijl's sulfur bacteria study and the use of the oxygen 18 isotopes that confirmed that the water was the sole source of the oxygen that is the byproduct of photosynthesis. That means then that water now has to become both a reactant and a product, because if it is a product in the chemosynthesis of the sulfur bacteria, then it must also be a product in photosynthesis if the two processes are parallel, with the H2S serving the same purpose as the water molecule. Therefore, water must also be a product because it's a product of the sulfur bacteria action chemistry. So that's the role of the sulfur bacteria and the oxygen isotope studies in figuring out the chemistry of photosynthesis. If your classmate says that the Calvin cycle is not necessary for the light reactions to occur, how would you respond? Well, what you could point out is that the light reactions require a source of NADP plus in ADP and PI in order to be able to continue to make NADPH and ATP. And those are only produced as a result of the Calvin cycle. So even though technically it could be possible for the light reactions to function in the absence of the Calvin cycle, in reality, that is not the case, because the Calvin cycle is the source of the NADP plus and the ADP and PI that the light reactions require to generate NADPH and ATP. So the two parts of photosynthesis, the light reaction and the Calvin cycle, even though they may appear to be independent, are in fact intertwined with each other. They're interconnected. and the NADPH and ATP and NADP plus and the ADP and PI are the bridges between the two sets of reactions. So that's how you could respond to that problem or that idea, or that proposal from a classmate. So given that we now have to have a source of energy to drive all of this, because we're trying to move electrons uphill from water, a low free energy, high electronegativity environment, to NADPH, a high free energy, low electronegativity environment, the exact opposite of what we're doing in cell respiration, we need a source of energy, and light is the source of energy. Everyone knows that. The question then becomes, what is the nature of light that allows it to function as the source of energy? Well, the first thing to recognize is that light belongs to a larger spectrum of energy called electromagnetic energy. And electromagnetic energy exists as waves primarily. That's how we normally think of it. So in terms of waves, you'll have, turn on the pen again, an electrical field that oscillates like this. And then at right angles to that is the magnetic field, which goes like this. And these are at right angles to each other. So the axes are at right angles to each other. And so you've got one of the two fields oscillating in one plane and the other oscillating at right angles in the other plane. And they move in that direction as waves. Now, when we start thinking in terms of waves, we think in terms of wavelength, the distance between two crests or two adjacent crests. So the wavelength would be measured from here to here, and that would be the wavelength. And we typically give the wavelength the symbol lambda. That's the lower Greek letter lambda, and that is the typical symbol for wavelength. So if in your outside studying you come across reference to lambda, that's referencing wavelength. So what's so important about the wavelength? Well, it turns out that wavelength of electromagnetic radiation is a function of the energy level. So the shorter the wavelength, the higher the energy level. The longer the wavelength, the lower the energy level. And so here you can see what is commonly accepted as the spectrum of electromagnetic radiation. At the long wavelength end, you have radio waves. Then as you move progressively from the right-hand end, the long wavelength end, towards the left-hand end, the short wavelength end, you find that the energy level gets progressively higher and higher. In the middle of the spectrum is that range of wavelengths that our eyes happen to be sensitive to, what we refer to as visible light or white light. And so it's referred to as white light because what we are detecting are all of these wavelengths in combination. We don't discriminate the individual wavelengths when we see white light. We're seeing all the wavelengths in equal amounts. But when you look at this diagram and you see this variation in color going from violet at the short wavelength end to red at the long wavelength end, and you see all the colors in between, the colors of the rainbow, what you're actually seeing is the different wavelengths that those colors respond to or correlate to. So violet, the short wavelength end, is approximately 380 nanometers in wavelength. Red, at the other end of the spectrum, the visible light spectrum, the longer wavelength, and therefore lower energy, is around 750 nanometers. In between are all the other colors, and each color reflects a particular wavelength. So when we talk about red or yellow or green or blue or violet, we're actually talking about different wavelengths of light that our eyes are sensitive to. And we have in our eyes 3 different color receptors. We refer to them as cones. And each of those different color receptors detects a different primary wavelength of light. Those primary wavelengths of light then can be combined together to get secondary colors, and then a secondary and a primary can be combined together to get a tertiary color. And that's what allows us to see the broad wavelengths of colors, or the broad spectrum of colors that our eyes are sensitive to. It turns out that plants are sensitive to the same wavelengths. So they can detect light from about 380 nanometers through to about 750 nanometers, because those happen to be the wavelengths that are detectable by most forms of life. Now, some forms of life, most notably insects and some birds, can see in wavelengths either side of the visible wavelength. So insects particularly can see down into the ultraviolet. Birds can see a little bit into the infrared, and some birds can see down into the ultraviolet as well. But the vast majority of life forms on the planet are sensitive to the same wavelengths that we are, for similar reasons, because of molecules referred to as pigments. It is the presence of pigments and their ability to interact with various forms of electromagnetic radiation that actually determines what colors we see. So how does that come about? Well, if we take a look again at these different colors, which, by the way, we see in a rainbow because of the ability of water to scatter the light, to split it up. The different wavelengths pass through water slightly differently, and that separates them out into their colors. This was first recognized by Isaac Newton back in the mid-1600s, in the 1650s. He used a glass prism, which is a triangular block of glass that has the same effect as water molecules in the air, and splits the white light up into the visible components. And what we now recognize is that our ability to see these different colors are the result of these different pigments. So why do you see something as red? Well, the reason why is because the red light is being reflected by the particular colored pigments that are in that material. So if we are seeing this as being red down here, there's a red pigment being generated, or a red light being generated in the case of the computer screen. And it's only the red light that is impacting your retina and your color receptors in your retina. And so you detect that as red. When we see the violet end down here, again, it is the violet light that is the only light that is coming from the computer screen, or something that you see as violet. Or perhaps we should think in terms of blue, because red and blue are 2 of the primary colors. Cyan is the 3rd primary color that we see by. And it's that combination of red, cyan, and blue, and the combinations of those that allow us to see the different combinations of colors that we can see. So why do we see something as blue? Because blue light is being reflected or being generated in the case of the computer screen, and it's only blue light that's reaching our eyes. When we see white light, or we perceive something as white, it's because all of the colors of the spectrum are being generated by the computer screen, or being reflected by the object that we see as being white. When we see something as black, nothing is being reflected. All the colors are being absorbed. So it's really all about what is being reflected by the object versus what is being absorbed by the object. And that's a function of the pigments, these molecules that interact with the waves of light to either reflect them or to absorb them. If the wavelengths are absorbed, we don't see that color. If the wavelengths are reflected, we do see that color. If all the wavelengths are reflected, we see that the object is being white. If none of the wavelengths are reflected, if they're all absorbed, then we see that object is black. And the other colors we see accordingly by what is being absorbed and what is being reflected. So the question then becomes, why are leaves green? And the answer is because the green light is the only wavelength of light that's being reflected. So that tells us something about photosynthesis. It suggests that green light is not effective, or is at least not absorbed, to drive photosynthesis. That red and purple violet light are the wavelengths that are primarily absorbed, because they're the ones that we don't see. We see green being reflected. Can we actually demonstrate that? The short answer is yes, we can. Now, it's the reason why you see leaves as green, because all the other colors of the spectrum are being absorbed in some fashion. Green is the one that's being rejected, either as reflected or it just passes all the way through. The others are being absorbed in the chloroplasts inside the leaves. We can demonstrate that this is the case by isolating chlorophyll from a leaf, and that's relatively easy to do. You can use something like methanol, or you can even extract it with alcohol. So you could use vodka or something like that that contains ethanol, and that would extract the chlorophyll. You could use rubbing alcohol. It's not quite as good, but it will still extract the chlorophyll. If you then put the chlorophyll in a machine called a spectrophotometer, a spectrophotometer allows you to pass light of very specific wavelengths through different solutions. So if you isolate green light from the spectrum and pass that through the solution, it passes all the way through, and you detect the presence of the green light on the other side with a light detector. And the spectrophotometer will indicate that it sees the green light passing through the solution. If you use blue light, or you could use violet light, or you could use red light, you could use yellow light, what you'll get is virtually none of it passing through. Why? Because all of that is being absorbed by the chlorophyll. So the green pigments, the green chlorophyll pigments, are what are absorbing this, absorbing the light. Now, this was actually first demonstrated back in the 1860s by a German biologist named Theodor Engelmann. And the result that Engelmann got is so cool and so perfect that even with all of our modern technology, with our spectrophotometers and all our fancy equipment, we really can't improve on Engelmann's result. So let's take a look at what Engelmann did. He took a filamentous algae, and the algae that's shown here is Spirogyra. So I'm not entirely sure whether it was actually Spirogyra that was used, because I can't find a copy of Engelmann's paper on the internet so that I can read it. But every diagram that I've ever seen shows Spirogyra as the algae, so I'm assuming that it was Spirogyra. And it forms a filament. Then he used a glass prism and sunlight energy, because there were no electric lights at this point. So he split sunlight into its component colors, just as Newton had done, and allow the different colors to fall on different sections of the algae. Then he used the observation that oxygen is a chemical byproduct of photosynthesis, something that was first demonstrated back in the mid-1700s by an English scientist named Joseph Priestley, who is actually credited with the discovery of oxygen through his experiments. Now, Priestley did not know that he had discovered a new element, oxygen, but he used the term oxygen because he had discovered that a plant produces something that revitalizes air that had been devitalized by burning a candle or by having a mouse sitting in the air, consuming something in the air and devitalizing it. So he'd put candles in glass jars, he'd put mice in glass jars and discovered that they couldn't survive in there indefinitely. But he had also discovered that a plant in light would revitalize the air so that it would support a burning candle or support a mouse. And he named this revitalizing factor oxygen, not knowing that it was actually an element. And he discovered that it was produced by plants in light. So he had discovered oxygen as a byproduct of photosynthesis without even knowing that that's what he was doing. But by the time Engelmann is carrying out his experiments, it was known that oxygen was an element and that it is a product of photosynthesis. Oxygen gas is produced by photosynthesis. So Engelmann simply said, if oxygen is being produced by the algae carrying out photosynthesis, it's going to accumulate in whatever wavelengths of light are best at driving photosynthesis. And I can demonstrate where that oxygen is being produced by using aerobic bacteria, because aerobic bacteria will always move to where there are high levels of oxygen. So we introduced aerobic bacteria into his experimental chamber and watched to see where they accumulated. And they accumulated in the purple-violet portion of the spectrum and in the red and yellow portion of the spectrum, and essentially avoided the green end of the spectrum. And if we trace a curve around this accumulation as outlined here, what you get is a curve that we call today actually, I'll write it. It's called the action spectrum. And it reflects the role of the different wavelengths of light in driving photosynthesis. And as I said, when we do an action spectrum of photosynthesis using our spectrophotometers, we get a curve that looks essentially exactly the same as Engelmann's. We can't improve on it. So it tells us that Engelmann got it right in figuring out that it was the blue-violet end of the spectrum and the red-yellow end of the spectrum that primarily drives photosynthesis. So the question then becomes, why? Why is it those wavelengths? Well, we know that it has something to do with the green pigments, the chlorophylls. So when we take a look at the absorption spectra of the individual pigments, and I'm going to move this out of the way so we can see the top curve for a moment. If we were doing wet labs instead of doing virtual labs, we would actually isolate these pigments using a process called thin layer chromatography so that you could see the actual pigments. And you would see chlorophyll A as a blue-green pigment. You'd see chlorophyll B as a yellow-green pigment. And then you would see this group of pigments called the carotenoids that are bright yellow in color. And we'd be able to isolate these on thin layer chromatography. And in fact, the technique of chromatography was specifically developed back in the late 1800s and early 1900s to do just this, to separate out the pigments from plant leaves so that you could identify the different colored pigments. Thin layer chromatography is just the most modern, easy to do version of it. There are a number of different ways that you can do this, but thin layer chromatography is what we would use in class, and it's a very neat way of doing it. And when you take a look at the combination of the individual absorption spectrum, so you see this big peak here. Oops, let me fire up the pen again, make sure that we've got the pen going. So when you see this big peak here in the blue-violet, it's the combination of the chlorophyll A, the chlorophyll B, and the carotenoids. And in fact, the carotenoids are what are contributing to this little absorption region in the green. So a little bit of green light is absorbed, but not very much because the carotenoids are able to do it. Then this peak down here in the yellow and the red is the combination of a second absorption peak of chlorophyll A and chlorophyll B. And you'll notice that this matches very nicely with what Engelmann was showing with his result. So we can account for Engelmann's result. We can account for the modern action spectrum of photosynthesis by looking at the combined absorption of the chlorophyll A and the chlorophyll B, and also the contribution of the carotenoids to the absorption in the blue-yellow, in the blue-violet portion of the spectrum, and then the combination of the chlorophyll A and the chlorophyll B in the yellow-red portion of the spectrum. And so now we understand why the action spectrum is what it is. It all comes down to the chlorophyll molecules and the carotenoids working together to maximize the absorption in those regions. Now, it shouldn't come as any surprise to you that these are the colors that are being favored. After all, the carotenoids are yellow. And remember, it is the color of the pigment that tells you what wavelength is being reflected. So the carotenoids would reflect the yellow, but they are able to absorb violet and blue. The chlorophylls are both seen as green. So one is blue-green, the other is yellow-green, which tells you that they are rejecting the green and, to some degree, the yellow and a little bit of the blue, depending on which one of the chlorophylls you're looking at. But because their peaks don't overlap, they complement each other, both in the blue-violet and the red, and the carotenoids also complement in the blue-violet. That accounts for the absorption spectrum. With very little absorption in the green, most of the absorption in the red and the blue-violet. So now we know what color wavelengths of light are the ones that are most effective at driving photosynthesis, and also which wavelength is the least effective. That would be the green. So when we look at chlorophyll molecules, and this is a diagram of the chlorophyll molecule, what we notice is this ring structure. It's called a porphyrin ring. And the porphyrin ring is where the light is absorbed. The electrons in here are very loosely held by the presence of these double bonds in the porphyrin ring. And that means that they can absorb, not only absorb energy, but those electrons, once they're excited by the absorption of that energy, can be captured so long as the chemical environment is appropriate for capturing those high energy electrons and extracting them out of the chlorophyll. Of course, if we say extract them out of the chlorophyll, we're thinking in terms of oxidation. So it's all about oxidizing the chlorophyll molecules by capturing the energized electrons as a consequence of capturing light energy. But we also anchor the chlorophyll molecules into the thylakoid membranes because they have a hydrocarbon tail, a hydrophobic region. So remember, hydrophobic wants to be with hydrophobic. We know that within the membrane, there is that large hydrophobic region as a consequence of the phospholipid bilayer. So this now provides us with an ability to anchor the chlorophylls into the thylakoid membranes to allow for the capture of light energy. It's a little bit more complicated than that, but that's basically where we're going with this. And the difference between the two chlorophyll molecules, chlorophyll A versus chlorophyll B, very subtle, but nonetheless very important. A more hydrophobic functional group, the methyl group in this location on chlorophyll A more hydrophilic functional group, the carbonyl group, in the same position on chlorophyll B. That subtle difference changes the absorption spectrum of the chlorophyll A versus the chlorophyll B. It's also what allows us to separate them using thin layer chromatography, because one is more hydrophilic, one is more hydrophobic. That means we can separate them using hydrophilic versus hydrophobic solvents in combination. And that allows us to separate out the two pigments in thin layer chromatography. So it's these subtle differences that account for the differences in absorption. Now notice also that there's magnesium in the center of the chlorophyll molecule. That magnesium is important in part of stabilizing this. The porphyrin ring actually is also present in hemoglobin. It's what holds the iron. So what's the difference between a porphyrin in chlorophyll versus a porphyrin in hemoglobin? The metal ion that's in the center. It also is the reason why they are different in color. It accounts for the green color of chlorophyll versus the red color of hemoglobin. And if you're a Star Trek fan, like I am, you'll know that Vulcans have green blood. And the reason why is because their hemoglobin has magnesium in the porphyrin ring. Yeah, people really do think about these things. And Star Trek was particularly good, in some areas at least, in terms of thinking about the science. And I've talked about Star Trek science before. Star Trek science is, and the writers for Star Trek have actually paid a lot of attention to the science. But I digress. Let's move on. So when we start thinking in terms of the behavior of light, we have to start thinking in terms of light in much the same way as we've been forced to think about electrons. Remember, we thought about electrons as both particles and waves. We could draw electrons as physical particles circling around the nucleus, or we could draw them as clouds of negative charge, waves of negative charge around the nucleus. Well, we have to think about electrons in exactly the same, or photons, or light in exactly the same way. We know that light consists of waves, the electromagnetic spectrum, but light and electromagnetic radiation also can behave as particles. And each type of electromagnetic radiation has its own particles, and these particles are called photons. And it's the particle nature of light that becomes really important here, because waves tend to be reflected from surfaces, but particles can collide with each other. So when we talk about light exciting electrons, it's really all about the particle of light, the photon, colliding with the electron as a particle, and there being a transfer of energy as a result of that collision. Now, remember, the wavelength of light determines the amount of energy. So there is a photon that has the energy of violet light, a photon that has the energy of blue light, a photon that has the energy of red light, da 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 da. And so the photons of purple and blue light are the most energetic, and the photons of red light are the least energetic. And that determines what happens to the electrons. I don't have a diagram, unfortunately, so I'm going to have to draw one. So you may recall, hopefully you do, that electrons exist in energy shells surrounding the nucleus. So here's a nucleus, and I need to draw in 3 energy shells so that you can understand this. So let's put an electron sitting here. If it gets excited by red light, so red light comes down and a photon of red light collides with the electron, it's going to get excited and it's going to jump out to here, because the energy of red light will give it enough energy, potential energy, to move out to this energy shell. If it gets excited by blue light, so here's a photon of blue light coming in, and that collides with the electron. Well, we know blue light has more energy. So the electron is going to come out here in its excited state. Now, the problem with being in your excited state is you don't want to be there. You want to come down back to your resting state or ground state. So for the red light electron, it's going to come back straight back down to here. And of course, it gained energy to get there. So it's going to have to give off energy. And we'll talk about what happens when it gives off that energy in a moment. The blue light, it turns out, does it in two steps. It comes down to the red light level, and then it steps down to the ground state. So it's a two-step loss of energy. Now, how does this explain the rejection of green light? Do I have green? Yes, I have green. So when green light comes in, this is going to get a little messy, so let me bring the green light in from here, I guess. When green light comes in, green light, remember, sits in the middle between red and blue. So the energy of green light would bring the electron out to here. But there's no energy shell for the electron to jump to, so green light gets rejected. So this is why it's red light and blue light that is absorbed, because of where the electrons can jump to in response to the collection or the capturing of that light energy. Now, if the chlorophyll is isolated out of the leaf and is in solution, then there's no way to capture the excited electron, and it jumps back to its ground state. And because both the blue light and the red light excited electrons eventually jump down from the red light level, both of them give off energy in the form of a photon of red light that our eyes can detect. And that gives rise to what is referred to as fluorescence. So what's going on here is this is a chlorophyll solution. I know it doesn't look green, but it is a chlorophyll solution. And it's absorbing light energy. The electrons are being excited, and they're jumping out to the appropriate energy cells, depending on whether it's blue light or red light that's doing the exciting. Remember, the blue light ones come back down to red light level, and then they jump, both the red light and the blue light jump down from there to ground state. And it's the giving off of the red light energy that gives rise to this red fluorescence. Fluorescent light bulbs do the same thing. They capture the gases inside the tubes, capture energy from the electricity passing through, get excited, and eventually the energy that they give off is given off in the wavelengths that our eyes are sensitive to, and we see white light coming out, or some variation of white light, depending on the type of light bulb and the gases that are inside it. Any type of fluorescent tube, so a neon tube that gives off different colors, or the advertising signs that have different colored lights, that's all about the gases and how they're being excited and the fluorescence that they're giving off. It's the same principle. Unfortunately, if this was the situation in leaves, we'd not be capturing light energy. So obviously, this idea of excited electrons coming back to ground state, not a good thing if we're going to carry out photosynthesis. So the chlorophylls have got to be organized in some fashion to allow for the capturing of these excited electrons. And that's where a complex of proteins and pigments called a photosystem comes in. So a photosystem, and I'm going to jump over those two slides. You can read those. You can download the slide set and you can read those if you wish. A photosystem is a complex of proteins arranged in what is called a light harvesting complex. So you can see that here. So these are also known as LHCs, or light harvesting complexes. Then we have the reaction center, which is where the energy is captured. And then at the base of the reaction center are a pair of chlorophyll A molecules called the reaction center chlorophylls. So these are the chlorophyll molecules that generate the high energy electrons. Now, that poses us with a problem, because on one of the previous slides that I jumped over, it says that there are some 200 to 300 chlorophyll molecules, both chlorophyll A and B, in the light harvesting complexes. So if only the pair of reaction center chlorophyll molecules are the ones that are generating the high energy electrons, the question becomes, what are all the other chlorophyll molecules doing? And it turns out that they have a special purpose. So the non-reaction center chlorophylls, and that's the shorthand for chlorophyll, CHL. I want to be able to write. And that's not going to do it. So hopefully you can follow that. Let me just put a red line to separate the two for you. So if we want to maximize the amount of light energy that we capture, relying on just the reaction center chlorophyll is not the way to go. We need a larger complex of chlorophyll molecules that capture light energy, and that's what the remaining chlorophylls actually do. And they pass that captured energy onto the reaction center chlorophylls, and they do so extremely efficiently. It's one of the most efficient energy transfers that we know of. About 90% of the captured energy is passed on to the reaction center chlorophylls. If you want a good analogy, think in terms of a satellite dish. The actual dish is capturing a lot of energy, but it's reflecting that energy to the little box on the end of the stalk in the center of the satellite dish, which is where the receiver amplifier is located that sends the signal on to your television or whatever you're capturing the energy for. It's a unique feature of the dish that it allows it to reflect it to a central point to capture, to increase the amount of energy that's being captured. And the light harvesting complexes basically do the same thing. So it turns out that there are two of these photosystems in leaves, known as photosystem 1 and photosystem 2. And each have their pair of reaction center chlorophylls generating these high energy electrons. Now, that's great that they generate these high energy electrons, but remember, if we don't capture them as high energy electrons, they're going to return to their ground state, and that energy is going to be lost. So also as part of the reaction center complex is an electronegative molecule referred to as the primary electron acceptor. And we know what these molecules are in some detail. It's not important that you know what they are, just remember that they do exist. And these primary electron acceptors, as electronegative molecules, are geared to capturing these high energy electrons. The electrons are further away from the nucleus than they otherwise might be because they're high energy. That means they jump out to an energy shell further away from the nucleus, which makes them vulnerable. And then you've got an electronegative molecule sitting right next to and within the reaction center. So when these high energy electrons are generated, they're captured immediately by the primary electron acceptor and removed from the chlorophylls as high energy electrons. And they are then moved on into an electron transport chain, as you'll see in a moment. So this is how we capture these high energy electrons. They are energized by the captured light energy. As they're energized and they move out to the higher energy shell, they're captured by these electronegative molecules, these primary electron acceptors, that remove them from the chlorophylls while they're still in their high energy state and allow the high energy electrons then to be moved through an electron transport chain till they are ultimately temporarily stored in NADPH. So when we think in terms of the two photosystems, we have photosystem 1, often referred to as P700, because its reaction center absorbs light best at 700 nanometers. And then ultimately a second photosystem was identified, and it's referred to as photosystem 2. And it's sometimes referred to as P680 because its reaction center absorbs light best at 680 nanometers. Now, they also have abbreviations. So photosystem 1 is abbreviated PS1, and photosystem 2 is abbreviated PS2. And so you'll hear me refer to PS1 and PS2. That is how in plant sciences we refer to them. Instead of constantly talking about photosystem 1 and photosystem 2, we'd rather say PS1 and PS2. So this is all terminology that refers to the same thing. So how do these photosystems go about capturing or driving the capture of high energy electrons? Well, it turns out that it requires the cooperative function of both photosystems to generate the high energy electrons necessary to reduce NAD plus to NADH. Along the way, however, some of that energy is siphoned off to generate ATP because there's more than enough energy captured by the two photosystems working together to not only energize the electrons to the point where they can reduce NADP plus to NADPH, but to also provide energy for making ATP. And in the process, oxygen is produced. So let's take a look at the way in which this is done. This is referred to as non-cyclic electron flow or non-cyclic photosphorylation, because we do wind up phosphorylating an ADP to make ATP. So there's where the phosphorylation comes from. Why photo? Because it's driven by light energy. So unfortunately, the role of the two photosystems is the reverse, or the functional role is the reverse of their name. So photosystem 2 functionally operates before photosystem 1 in this scheme called the non-cyclic electron flow, also known as the Z scheme. Why Z scheme? Well, if you turn your head on the side like this and look at it, you can see that the flow of electrons traces out what looks like the letter Z. I'm not sure why they, if I'd been looking at it, I would have said it's the N scheme, but because it looks like the letter N when you have your head upright. I'm not sure why they said turn your head on your side so it looks like a Z. But historically, that's what it's been talked about, or it's been labeled as. So when I talk about this, I like to start with P700 because that's the ultimate source of electrons for reducing NAD plus to NADPH. So let's start here. So we start with the electrons, and I'm going to do this with red to start with at least. So we start with our P700 being excited by energy captured from light, either by light activating P700 directly or through transfer of energy from the antenna complex to P700. Irrespective, it generates two electrons, two high energy electrons that are then captured by the primary electron acceptor. Remember, it's more electronegative, so it captures the electrons. It passes that off then to a protein called ferredoxin. It's got iron in it, hence the ferri part of it. So this is an iron-containing protein called ferredoxin. And the pair of electrons are then transferred to an enzyme complex called NADP reductase. And you should recognize this as an enzyme because it ends in ASE. Okay. So the electrons are then utilized by the enzyme to reduce a molecule of NADP plus to NADPH. That leaves the P700 chlorophylls and this is important. When you lose the electrons from the chlorophylls, they are now oxidized. The primary electron acceptor gets reduced. As a consequence, when it gives off its electrons to ferredoxin, it gets oxidized, ready to capture more electron. Ferredoxin is reduced. It passes its electrons on to NADP reductase, which reduces it, leaves ferredoxin oxidized, then the enzyme reduces NADP plus to NADPH, But the end result is here, the P700 chlorophylls are in their oxidized form. Why is that important? Because oxidized chlorophyll cannot generate more high energy electrons. Only the reduced chlorophyll can do that. So we need to reduce the P700 chlorophylls back again to their reduced form, but that requires electrons. So the electrons for the reduction of the P700 originate over here in P680. So when P680 gets energized by light energy, it also produces a pair of electrons. Those electrons are captured by the primary electron acceptor and they are passed on to a mobile protein called PQ, which is hydrophobic. So it's moving around inside the membrane and it passes its electrons onto a complex of proteins called the cytochrome complex. And the cytochrome complex does in chloroplasts exactly what the electron transport chain protein complexes do in mitochondria. It takes hydrogen ions and moves them through the complex from the stroma to the thylakoid space. Oops, that's not what I wanted. We want to see that. So the hydrogen ions are being mobilized into the thylakoid space to create a gradient of hydrogen ions. Sounds a little bit like a mitochondrion, doesn't it? In the thylakoid membrane, there's an ATP synthase that can then process the flow of hydrogen ions back from the thylakoid space to the stroma and use that as an energy source to make ATP. And here's the ATP being generated as a consequence of that. When the electrons exit the cytochrome complex, that results in the hydrogen ions being released in the thylakoid space, but the electrons are passed on to a second mobile protein called plastocyanin, or PC. PC passes the electrons on to the P700, which reduces P700 back to its reduced state again. So it can generate more high energy electrons for the reduction of NADP plus to NADPH. But now we've got oxidized chlorophyll over here in P680. We need to reduce it so it can continue to provide electrons for reducing P700. And this is where water comes in. The water is split by a complex of proteins and metal ions called the water splitting complex, which also sits in the thylakoid membrane adjacent to photosystem 2. And as photosystem 2s lose their electrons, the water splitting complex provides electrons to reduce the P680 back to its reduced form again. And we think we have a reasonable idea about how it goes about doing this. It splits the water molecules by storing electrical charge until there's enough electrical charge to split a pair of water molecules. Why a pair of water molecules? So that the oxygen can be released as oxygen gas, O2, rather than as individual oxygen atoms, because individual oxygen atoms are highly reactive and very damaging. And for plants, the presence of the oxygen is quite a problem. That's why they have those peroxisomes. And so being able to release the oxygen as oxygen gas is kind of important, but that takes 2 water molecules to create oxygen gas. So we think the water splitting complex is simply storing up electrical charge until there's enough to split 2 water molecules. The oxygen gas is then released as the byproduct. The hydrogen ions contribute to the generation of ATP. So I'm going to draw an arrow over here. So those hydrogen ions from the splitting of water contribute to the generation of ATP as an energy source for the Calvin cycle, the reduction of carbon dioxide to organic molecules. So this is non-cyclic electron flow. It generates the ATP, it generates the NADP plus that's necessary for the reduction of carbon dioxide to sugar. However, it's not the only utilization of light energy, because it turns out that the Calvin cycle actually requires a little bit more ATP, about half as much again, ATP as it does NADPH. And the amount of ATP to NADPH here is about 1:1. So if you're going to make extra ATP, it would be good to have a second way of making ATP. And as it turns out, there is. And it's called cyclic electron flow. So we've got non-cyclic, we've got cyclic electron flow. And in cyclic electron flow, the beginning looks very much like what goes on in non-cyclic. Photosystem 1's reaction center, the P700 gets activated. A pair of electrons are generated, and they are captured by the primary electron acceptor, which passes them off to ferredoxin. But it turns out that ferredoxin is an equal opportunity donor of electrons. It doesn't care who it donates them to, as long as it's more electronegative than it is. And it turns out that the cytochrome complex and the NADP reductase are both more electronegative than ferredoxin and both sit in the thylakoid membrane. So which direction the electrons go really reflects which of these two complexes does ferredoxin encounter first, NADP reductase or the cytochrome complex. And in some locations in the chloroplast, there is virtually no NADP reductase, but there's a lot of cytochrome complex, which means that the photosystem 1s in that area are dedicated to cyclic electron flow and the production of ATP. And that's something that we've only just really started to realize in about the last 15 years or so, that there are photosystem 1s that are totally dedicated to cyclic electron flow and therefore totally dedicated to making ATP only. And so the electrons from the ferredoxin pass into the cytochrome complex, contribute to the buildup of hydrogen ions, which means that you're contributing to the buildup of ATP or the accumulation of ATP without producing NADPH. So this is where we are now very confident the extra amount of ATP is coming from that's necessary for the Calvin cycle, as you'll see. But the presence of this cyclic electron flow also tells us something about the origin of photosynthesis. The reason why we say probably represents the early origin of photosynthesis is that we know approximately when in the history of the planet oxygen started to become a significant part of the Earth's atmosphere, and that's about 2.5 billion years ago. We start to see a buildup of oxygen to around 10% of the level that we have now. We actually have about 20% oxygen in the atmosphere, so we're talking about an atmosphere that contains 2% oxygen. And you might think, well, that's not a lot. Well, what you have to bear in mind is that up until this point, there was none. And so in terms of oxygen being a byproduct of photosynthesis, early on, whatever photosynthesis process that might have occurred was not producing oxygen. That would come much later. Around 3.5 billion years ago is when we start to see the beginnings of what we refer to today as oxygenic photosynthesis, photosynthesis that produces oxygen. But it would take a while for it to start to build up oxygen into the atmosphere for a number of reasons that we don't need to go into right now, but that is the case. But something must have preceded oxygenic photosynthesis, and we think this is it, because this is clearly an advantage to any bacterium that does it, because it's a way of making ATP by just lying out in the sun and capturing light energy. What reinforces this is that we now recognize that photosystem 2 represents a mutant of photosystem 1. It's not identical to photosystem 1, but it is similar. And we can now account for the origin of photosystem 1 as a product of mutations associated with the genetics of photosystem 1. So photosystem 1 almost certainly was the first of the two photosystems to appear in the history of photosynthesis. Photosystem 2 comes along later, which allows for oxygenic photosynthesis. But in the meantime, organisms are cashing in on the ability to capture light energy and make ATP, and there are still some bacteria that do this today. So we think this gives us some clues as to where photosynthesis came from, just as glycolysis gives us some clues as to where cell respiration may have originated in the absence of oxygen. So what we now have is the ability to create chemical energy in the form of ATP and reducing energy in the form of electrons in NADPH, using electrons ultimately sourced from photosystem 2 or from water via photosystem 2 and the electron transport chain, photosystem 1, in order to have high energy electrons for reducing NADP plus to NADPH. So one of the other things that we also need to bear in mind, and I've already pointed this out, but let's point this out again, the way in which we make ATP in mitochondria and chloroplasts is identical. The only difference is where the complexes are located. So in mitochondria, the electron transport chain and the ATP synthase are in the inner membrane, the Christie membrane, and the two compartments that are separated by the membrane are the matrix and the intermembrane space. In the chloroplast, the equivalent membrane is the thylakoid membrane, and the two compartments are the stroma and the thylakoid space. Everything else remains the same. And in fact, the experimental evidence was first obtained in chloroplasts or from chloroplasts. Making ATP. In the dark. And this is actually quite a cool experiment because given that the light reactions are the source of the ATP, and ATP is typically generated in chloroplasts through the capturing of light energy, to have chloroplasts make ATP in the dark means that the actual production of ATP must be tapping into the light energy in some fashion, but the light energy is not the direct way in which the ATP is being made. And so chloroplasts were manipulated in order to be able to make ATP in the dark in the absence of light energy in terms of changing the hydrogen ion concentration in the thylakoid space by adjusting the pH. Remember, pH is a measure of hydrogen ion concentration. So the researchers simply altered the pH of the thylakoid space, not a terribly difficult thing to do. And in response to increasing the hydrogen ion concentration in the thylakoid space, these chloroplasts that were being kept in the dark started generating ATP. It was the flow of hydrogen ions that was actually driving the synthesis of ATP. And that the chloroplast could do this in the dark meant that the flow of hydrogen ions was it. And therefore, given that the two mechanisms in chloroplasts and mitochondria were absolutely identical, it must also be the case in the mitochondria. And that led to the awarding of the Nobel Prize for Chemistry in 1961 to a man named Peter Mitchell, who had first proposed that it was the flow of hydrogen ions generated by an electron transport chain that was responsible for generating ATP. It was the ability of chloroplasts to be able to do this in the dark by simply manipulating the hydrogen ion concentration that demonstrated that, in fact, Mitchell's idea was absolutely correct. So that's one of the 3 or 4 Nobel Prizes that have been awarded to our understanding of how the ATP synthase makes ATP. That one for Mitchell was one of them. So we now have an understanding of how to make ATP. It's exactly the same in chloroplasts and mitochondria, no real mystery there. So this is the diagram that brings the light reactions together. We have the two photosystems linked together by the electron transport chain, the mobile proteins PQ and PC, plus the cytochrome complex moving the electrons from the energetic electrons from photosystem 2 through the electron transport chain to photosystem 1, where they are re-energized and moved through the ferredoxin to NADP reductase and used to reduce NADP plus to NADPH. Along the way, hydrogen ions are transported across the membrane through the cytochrome complex as the electrons pass through it. Hydrogen ions from splitting water as a way of providing electrons for replacing those that are moved from photosystem 2 onwards. Also contribute to the hydrogen ion gradient to generate ATP. Remember, we have linear electron flow or non-cyclic electron flow producing both ATP and NADPH, and then we have the cyclic electron flow involving just photosystem 1 that contributes only to the production of ATP to give us the extra ATP that we need to drive the Calvin cycle. So now we have our sources of energy, the NADPH having the reducing power through the pair of electrons that it holds, and chemical energy in the form of ATP, both of which are now going to be required for the reduction of carbon dioxide to sugars. So time for another break with a concept check, and we are going to take a real break here, and I'll meet you on the other side of the break to answer these two questions. Which color of light is or are least effective at driving photosynthesis? And what is the initial donor for the light reactions? And where do these electrons eventually wind up? I'll see you on the other side of the break to answer these questions. Yeah, so let me stop the recording here.